Sure. Thalassemia is a group of inherited conditions that prevents the bone marrow from producing enough healthy red cells. This leads to low levels of hemoglobin and can cause anemia. Left untreated, anemia can lead to severe health problems. And one of the key features of this is the tendency to accumulate iron in the body, and it is known as one of the iron-loading anemias. A key mechanism that controls the level of iron in the body, a protein called hepcidin, which is the key regulator of the body's iron. And what this protein does is to control distribution. And what happens in patients with thalassemia is that because of the iron overload, the levels of hepcidin are inappropriately suppressed, so that they tend to accumulate iron, particularly in the erythron, where the red cells are developing, but also in other organs such as the liver and potentially also the heart. So the idea of modulating hepcidin or changing the levels of hepcidin is to reduce the level to restore the normal distribution of iron and take it away from the developing red cell. We do this by interfering. We're working with a technology called sRNA, which is small interfering RNA, and that interferes with the message going between the gene and the protein, the messenger RNA, and causes it to suppress the action of a repressor of hepcidin. So the mechanism of action of the drug is actually increase the levels of hepcidin and reduce the effect of iron overload on the red cell. So SLN124 is currently in phase 1 studies and also studies in patients. We reported very nice data from our healthy volunteer study last year, which suggested that the mechanism of action is working how we expect it to, and it replicated what we'd seen in preclinical models. Currently enrolling patients with alpha and beta thalassemia in a single dose rising study. Having the data from that in the third quarter of this year, and we anticipate that we will replicate what we saw in the healthy volunteer study with elevations in hepcidin, which is what we're trying to do, and reduction in serum iron, which is the key component that we're trying to control. And animal studies have shown if we produce that sequence of events, then we have an effect on anemia, which should help in terms of blood transfusions. So we're looking forward to seeing the data in patients. They obviously have abnormal iron distribution, and we hope for equally positive results in the patients.